Okay, so we can start. So, good morning to everybody. I hope that you can hear us on Zoom well. Somebody can say something, it would be great. Yeah, okay, great. So today I will give a short presentation about the multilingual stakeholder survey we did in the Biodiversity Genomics Europe project, together with a colleague of Christian De Gatti, which is not with us today, unfortunately, and also in collaboration with the social scientists. So what was the main aim of the survey? What was we planned to do with the survey? It was a really short one, not more than 10 questions. We tried to get a little bit of insight about the stakeholder knowledge in the field of the biodiversity genomics, and also tried to just briefly identify the possible need which we can work as a scientist in the future to get a better involve them and engage with them with the stakeholders in the field of the biodiversity genomics. So I will just give you a brief introduction, because if we are discussing about the biodiversity genomics, it's always good to remember what exactly is a biodiversity, despite it's coming from the Greek word which means life. We know that today we recognize the biodiversity mostly to the species diversity. All of us can very nicely can recognize the different species. We know that there are species from the tiny bacteria to the, I don't know, very large species like tree in the forest, or I don't know, mammals, birds, and any other taxonomic group. Even the lay public can really recognize the different species. But we shouldn't forget that there are two other levels of the biodiversity which are important, and that are ecosystem diversity and genetic diversity. And coming to the genetic diversity, we are coming very close to the genomics. So genomics, which try to understand how the DNA molecule, so which is, we always said in the research on genomics that this is a remarkable molecule, it's an informative molecule because it has a code of life, which is written just with the 4 letters, adenine, thymine, cytosine, and guanine. And we can say very in simple way, if the biodiversity is the story of the life, then the DNA is the language this story is written. And of course, then you can use the genomic to understand better this language. So we can say that one way genomic is like an X-ray vision of the biodiversity, because we can understand which species we are dealing, how do these species evolve, how do these species maybe are going to adapt, or how are going to be affected maybe in the future by the different humans activities which can influence, of course, also the population health and the species survival. So in the Biodiversity Genomics Europe project, we had this meeting for 2 days here in Porto, and we have a various case studies which was representing the possibility of use biodiversity genomic in conservation and management, and also in bioeconomy. This project put together 34 institutions across 20 different countries, but we must stress that many other countries' institutions were involved through the more than 30 different case studies. So it's a much broader approach to the whole European area, I would say, to the European countries. The Biodiversity Genomic Project is putting together 2 networks. One is ERGA, which is a European Reference Genome Atlas. ERGA is a European node of bigger global initiative, which is Earth Biogenome Project. And another network, which is also part of the project, is EYEBOL Europe, which is a part of the global network, which is called EYEBOL. So what exactly are those 2 networks and the researchers, which are also part of this Horizon project, which will end next year, were doing? The EYEBOL Europe is focusing on the DNA barcoding. What is exactly DNA barcode? Is a short molecule of the DNA, which is specific to each species. So we can recognize, we can identify, we can distinguish between the species using the short part of the DNA, which is a part of the each species genome. So we can detect and monitoring then the species in the nature. We can discover even the new species. And what we heard recently, the DNA Bank, eyeball Europe, and together with the eyeball, they discover more than 20. They describe more than 12 million species until this year. Other part of the project is connected with ERGA and with the reference genomes. So how do we define the reference genomes? We define them as a high quality genomes, which is the total DNA of the species. And it's having this really really precise information about the genomes. It really helps us to understand the species adaptation, because we can study interspecific genetic diversity, but we can also understand the better improve the science-based management and the science-based conservation by using the genomic data, because we can really see how the population evolve and how there are possibility to adapt in the change in the environment are happening now or is going to be happen because of the issue are going on. And many of those issues which we are facing today are also consequence of the anthropogenic influence and the human activities. So what we did, why we did this survey, the survey which includes more than 700 stakeholders. I will show you today results of including about 470 response. There are more of them we need to analyze, but it's quite a huge work to be done. So at the end, we will have more than 700 or even 800 response. We try to establish the baseline for the stakeholder knowledge. So we should understand what are the knowledge of the stakeholders and the need across the Europe. We try to identify just a briefly identify the possible barriers which the stakeholder recognize when they start to engage with the researchers. And at the end, we also try to get a little bit more information if the stakeholder would be interested in the training or some additional outreach activities which can bring biodiversity genomics close to them. So to beginning this work, we need to first identify our stakeholders. And this was a long way in the biodiversity genomics, especially in ERGA, because at the beginning, when we start our work, many of the scientists who were part of the network were not aware of the stakeholders and the people who can use, or the institution who can use their results. And it was a few years of work when we tried to identify and we tried to map at least some of the stakeholders, possibly not all of them, which can use and can be more involved in the biodiversity genomic research, and they can use those data also for their daily activities to better conserve, to better manage the population or the species. So we do identify different categories or different type of the stakeholders from the National Environmental Ministry and Agency, from the protected area, different NGOs and foundation, even the industry partners which are connected, for example, with the bioeconomy or even with the wild health approach, then different museum, herbaria, biobanking. We also have initiatives when we include citizen scientists, local communities, not as a just collection of the samples, but also they were actively involved in the interpretation, and of course, agriculture, forestry, and fishery agencies. But I would like to stress one important aspect. It's also we focus on the university and institute and of course the research infrastructures, because we know that in many cases we have a different area of research and different field, and some of those fields are not even familiar with what we can do in the genomics. So even those scientists has to be a little bit more informed and involved in the biodiversity genomics application. So with the help of the social scientists, because we would try to do the survey really neutral and easygoing, because it's very difficult, because we need to translate those survey into the 17 European languages. So you can see here all of the language we translate the survey and also the participation. But as I said before, the participation of the different stakeholders, it will change when we do analyze the all data. But it was quite a work of course, the people who help us to do the translation, and of course, after that, try to distribute the survey across the different countries, because we know that the national languages are really important because English is not so common in the stakeholder area. So just to show you a little bit of what we found until now. So one of the questions was, how do the stakeholder have some knowledge about the biodiversity genomics? And you can see that less than a half, they answered that they have this basic understanding. Some of them heard of it, 1/3 of them a little bit less heard of it, but they are not familiar. But we also have still group of the stakeholders which are never heard about the biodiversity genomics. Of course, we have also part of the stakeholders which are already involved, and those have quite knowledge in the area. Then the next question was if they, because we are in ERGA discussing quite a lot about the quality reference genome and how the reference genome is important baseline to do any additional genomic analysis and observation. So we asked them what is the reference genome, and you can see that majority of them already know what is a reference genome, but still some of them were not precise in the answer. So even there we have an answer that they think that there are genetically modified organisms, or that just we are using genome to predict environmental change. So there are still some lack of knowledge in this direction, even though most of them are very knowledgeable about the reference genome. Then one of the question we were also interested in is, are the stakeholders were already contacted or being contacted by researchers? So do they already contact researchers working in genomics, because they have some need, and only the genomic information can answer the question they're dealing in their daily work. So the question, how many of them were contact already academic researchers? You can see the more of the half of them already been in contact with academic researchers, and probably also have some ongoing active conversation and communication. But still there is a quite a high number of percentages, I would say, of them which were not in contact with the researchers, and they do not know what the researchers in biodiversity genomics are doing. And there was the opposite, which it was really surprise for us, because as the researchers, we always think that we do enough in the communication with the stakeholders, and that we quite communicate with them. You can see that the stakeholders replied that less researcher contact them comparing to when they contact the researcher. And we do not expect that this is going to change even when we increase the data set of the analysis. And of course, many of them were not contacted, even almost half of them were never contacted by a researcher in biodiversity genomics who responds to the survey. And then, of course, we also ask them if they are satisfied with the communication they have. And you can see that half of them, they were fine. Half of them, a third of them were very satisfied, but we still have also not satisfied people and not so very satisfied. So this is the thing we should work as a researchers in the future in this area. And then in the end, we just try to ask the stakeholders a really simple question. Do they think that some kind of portal or any kind of tool to exchange information, of course, in the national language, it's always meaning in their language, would be helpful for them? And you can see that half of them, they said they think that this kind of way of communication, because we are now in the digital society and we communicate quite a lot through the web, could be also quite a helpful way. But there are also some of them which are not pretty sure if this could be helpful. Probably those are those stakeholders who were never contacted by academic researcher. We need to analyze this data more in detail to see if there are some kind of correlation with these two answers. So just to summarize at the end, what we see, I'm not going through the all answers. These are just to give a brief insight what we found until now. So what we really see that like engagement of the stakeholders is always complex. It's not something it's easygoing in any field. Especially, I think that in the biodiversity genomics is even more complicated because it's a quite complex way of analyzing the data and to try to simplify those data, like we said in biology, in the black box, and try to output the results in the way everybody would understand. So these are the communication is the key aspect. And these are also something we would like to discuss today after my presentation. One of the main questions is the communication should start before we start the genomic project, or we should start communication after we already have the idea about the genomics, because we realize that in many cases, researchers start a project and at some point of time, they realize that maybe those data can be used for the stakeholders, or maybe the opposite way is it's more acceptable. So this could be really important with the continuous engagement and also for additional benefit sharing with the stakeholders, which are becoming quite important. So what we are going to do the future in the biodiversity in ERGA and also in the BG project, we will work on the publication and the way of publication that I would say that can be understandable to the stakeholders. We will also map all of the stakeholders. They already expressed their interest or they were already identified in our ERGA website. So it will be like interactive map. So each of the representative of each country can go there and see which are the stakeholders recognized in the countries in each country regarding in our research. So this is something we would like to do as soon as possible. And of course, one of the ideas is also to develop this web portal and the communication app to easier communicate with the stakeholders in future. So at the end, I would like to thank two additional persons, the social scientists, and of course, Luisa, which is with us today, to help us with the work and to prepare this presentation. And that's all from my side. And I think that we can now open the discussion.